Make sure we're recording here. Make sure, and it's working. Okay. All right. So you guys should be able to see my screen here. So this is a recording for, which will be like an instructional on how to generate your lab report. Okay. So I'm going to have a lot of windows open. I'll be doing a lot of clicking back and forth as I go between different files. And just as a something to note, so this lab report carries a lot of weight for your final grade for this class, and it is a significant project, right? So you're going to need to at least give yourself a couple weeks to do this report. The main reason for that is because some time is going to be spent working with your group on generating all of the tables and figures, and then time is also going to be spent on your own generating the rest of your report. So, you need to give everyone else in the group ample time to kind of work together to generate the results. Okay. Let's go ahead and get going here. So if you go to Canvas, there's a whole lab report module there. And it has a lab report guide, which is basically what I'm going to be going through step by step on how to generate your report. And then there are also 2 manuals that are going to be referenced as I go through that guide. So let's go ahead and open that up here. Also, as I'm referencing it, so be sure that you're looking at the right manual, manual 1 or manual 2. The reason we have 2 manuals because I've pulled from 2 different exercises that Bio-Rad does. The first one is just the SDS page module where you stain with Kamasi and then you do a proteomic profile. And then the second manual expands on that first lab and includes a Western blotting module. All right, so we went ahead and did both. So your lab report is going to include both. As such, I'm referencing both manuals. Okay. All right. So, your report is going to have an abstract, an introduction, a materials and methods, a results section, a discussion section, and a literature cited section. Okay. So everyone's lab report is going to be different except for the tables and figures in the results section. The tables and figures in the results section, everyone in your group is going to have the same exact tables and figures. So you need to work together with your group to generate those tables and figures. And then once each of you has a copy of them, you go off and you put those in your results section, and then you start writing up your lab report. All right. So there's additional writing in a results section besides just the tables and figures, and I'll get into that in a little bit. But tables and figures are the same. Okay, so the first thing you need to do is decide as a group what data you're going to use. Now, I've given to you here some kind of canned data. It looks pretty good. The blots are clean. All the lanes work. No problems with the gels or the Western blot. You're welcome to use that. And in fact, I'm going to use that as an example when we're kind of generating our standard curve and how to look at generate the different tables and stuff. All right, I think it should be pretty straightforward to do. We used 5 fish samples like we did, but the fish, there was some difference in the fish samples. So if you're going to use this data, you have to go back, I think, and fix one of the fish samples. I don't think we did sturgeon this semester. I couldn't get any sturgeon, so you have to print off another fish data sheet and fill that out for the sturgeon. I think these are all the same otherwise. Okay, now if you want to go ahead and try to use your data, that's fine. I'm good with that too, as long as you can. So some of the groups, it didn't work. One group here, the SDS page didn't work, probably through no fault of their own. I think this was a problem with the equipment. So if you want to try to use, class generated data, that's fine. Just grab a gel from another group. That's fine. Okay. For the SDS page, you guys did pretty good here. Only 3 groups work, so you can use those. For the Western blot, though only 1 group worked, you'll have to use this result from group 3 for that. Now, if you choose to use the class-generated results and you do it, successfully, like you generate the tables correctly and you're looking at the right bands and it's making sense, I'll give you 3 extra points on your lab report, okay? That's not a lot of extra credit, though, and I think it's going to be more difficult for you to generate data on your own results, actually, just because, the lab, because as I'm going through and explaining it, it's going to make, more. I'm going to use this as an example and whatnot, but it's not too bad. Like, I want to encourage you to try to use your own results, and that's why I'm kind of offering 3 points extra credit. Okay, so you need to agree as a group on what you want to do. You want to try to use your own, or you want to do the canned results. Okay, remember, there were 2 gels run, right? One was stained with just Kamasi, and one was then the gel proteins were transferred to a piece of nitrocellulose, and that was probed for myosin light chain. Okay. Again, this is the Proteomic Lab Report Guide. All right, you have to turn in 2 copies of your report by the due date. So 1 copy you're going to digitally upload, which is going to do here. And the reason for that is so that I can get feedback from the plagiarism and AI detection software. And then the other copy you are going to print out and bring to class, stapled, please, okay? And bring it to class to turn in. That is the copy that I'm actually going to grade, right? Because I need it in front of me. I need to write on it and whatnot. Okay. Do not use AI, Grammarly, plagiarism, do not cheat on this report. The plagiarism detection software is very good. It will pick it up, and I have also been grading these lab reports for a number of decades, and I know what to expect if you guys actually write them. So just don't mess with this. There's not a lot of time left in the semester. I'm not going to be able to give you an opportunity to rewrite it. There's just going to be a serious grade penalty for it, and it's going to affect your final grade. Okay, so just don't do it. Write your own lab report. In general, students have found generating this report and this exercise kind of interesting. If you think about it, it's kind of cool. We've done a very, very simple procedure here, just SDS page. You've done it twice now. You did it once with Rubisco and now once with this. And you can generate substantial information from that, such as generating a proteomic profile. So, use this as a learning opportunity. This is going to be very heavily tested in your final exam. So getting into this and understanding it is your goal. Okay. So 2 copies to turn in. All right. So these are the 5 species that I'm using in the sample data. Now, remember, I couldn't get sturgeon, so you're going to have to replace this with whatever fish we did. I forget. So I went over that in class. All right. So 2 manuals I'm going to be referencing here. Manual 1 is just the SDS page Kamasi. Manual 2 is the Western blotting stuff. Okay. So when I reference a page number, be sure you're looking at the actual page number, not the page for the PDF. Okay. So like this is not page 7 here. This is page 1. That's the page number that I'm referencing. For example, in Student Manual 1, I want you to read pages 33 through 73. That's a lot. But I want you to read it and understand it. And I want you to do the focus questions on lessons 1 through 3. So if we scroll down to page 33, I'm going to open this up in a different thing here so I can use it easier. So if I go down to 33, well, this is the wrong file. This is the right file. Wait, is this number 2? That's 2, I'm sorry. All right, bear with me, lots of files. There we go, 33. Okay, I want you to read all this stuff, right? Understand it, And you will see there are some focus questions. Here we go. Is this focus question? Yeah, focus questions that start coming up. I want to do all the focus questions. All right, the answers are in the back of the manual. Okay, it's not you getting it right or wrong. It's you understanding this information because, like I said, in order to write this lab report, it's going to tell a story. You need to understand the story that's going on here, and you're not going to understand it unless you read and do what I'm telling you right here, which is getting into these questions and answering them. Okay, so that's the first thing you're going to do. All right, and then the data sheets. Now, you have already done this, okay? So hopefully you've already done this too. But, each fish gets a data sheet, and you need to complete it for each fish, and the instructions on how to complete it are given on those pages. An example of one of them is given. Okay, that's one of your assigned fish, so you have one to begin with. And the text in these pages here is going to go over like how you're going to make a prediction into how close these different species are, in terms of like, the tree of evolution, right? How close would they be based on this information that you're getting in the fish data sheets? I propose that the salmon and the catfish are going to be very, or the salmon and the trout are going to be very similar because they come from freshwater stream, things like that. So you need to make your own predictions, and filling out those data sheets are going to give you information to make predictions. Generally, this is your hypothesis, like how these predictions are. Okay, so what these predictions are the basis of your report's hypothesis. Okay, there are also some questions, page 53 to 54 in Manual 1. 53 to 54. Yep, sorry, more focus questions. All right, just be sure you're answering those. And so these questions, the reason that you need to do these is because, when you're writing up your discussion, what are you going to write about? Well, these questions are going to guide you into seeing what the important points are for this lab exercise, and it's going to give you some ideas on what to write about in your discussion. All right, your discussion is not just, my blot didn't work, and because I could, and to fix it, I need to change the temperature. No, this is, you're getting into some real biological concepts here, and these questions are going to help guide you into the stuff that you need to include. Okay. All right. So let's go through his here. Okay. So after you have those 5 data sheets, you're actually going to create a table. I have to fix these table numbers. This is table 1. I knew I was right about that. So you need to create a summary table. Right? This is going to be a big table, and it's of your own design within your group. You guys can do it together. Where you're going to summarize all the data from those 5 fish sheets into one nice little table that you can kind of look at and really start to easily make your predictions. Okay, that's going to be the first table in your lab report, and that table is going to be the same among all individuals in your group. I'm just going to fix something real quick, guys, because I have a little error on here. That's table 1. This is, table 2, table 3. Okay, maybe there wasn't an error. There wasn't an error. Table 1. Yep, okay, I didn't have a mistake. I'm sorry. All right, so that's your first table. It summarizes all the results of those fish data sheets, these things. Okay, it's be a pretty big table, probably take up almost half a page, that table. So also that 6-page page recommendation, it doesn't include figures and tables. Okay, so it's just writing. That's actual writing. All right, so that's your first table. All right, now we're going to get into how to generate the other figures and tables. Okay, so first you're going to have two figures, right? Figure 1 and figure 2. These two figures are going to be these guys, right? Now, if it's your actual data, then it's your actual data. And, you know, you're going to have to label, if you're using your data, you're going to have to go in and label these lanes and label the molecular weight marker. Understand, I believe this is a different molecular weight marker. It is, yeah. This is, oh no, it's not. Actually, this is the same. We'll take a close look at that in a little bit. But be sure that you label your molecular weight marker and label the lanes. But this is going to be figure 1 and figure 2, okay, in the lab report. Now, important side note here, tables are going to have a descriptive title. It's going to say table 1, and here's your title. And then it's going to have descriptive column and row headings. And that's it. Figures are going to be whatever image you generate, okay, and a descriptive title, so figure 1 title, and then it's going to have a legend, okay. Now this is a crappy legend here. This is not what your legend is going to be. A legend provides some key insight into the figure. You know, for example, you know, fish muscle was run on a this percent, 12% SDS page gel, and the gel is probed with Kamasi so that different you know, protein profiles can be generated, et cetera, et cetera. Okay, so legends contain significant details for the figures that are needed so that your reader can see the figure in the legend and understand what he's looking at. Tables don't need those because they already have descriptive column and row headings, and they don't need a legend. Okay. All right, so that's figure 1 and figure 2. Where are we here? Right here. Okay, now I'm going to go over how to do this to generate the standard curve, because it just seems to trick up a lot of students. But instructions are provided here on page 75 in Manual 1. So that's what we're doing here. Okay. All right, so I'm going to go ahead and pull up what I'm doing here. Now, what I did earlier, I went ahead and grabbed group 2s, and I, you know, kind of worked it up. And then I also grabbed the image of the CAN data, the SDS page CAN data, and worked that up as well. Okay, so this is the in-class data, which I'm actually going to delete here in a little bit, and I'm just going to work forward with this. I just want to check and make sure that the molecular weight marker is the same, 37, 25, 20, 15, 10. Right, it is. Okay, so how do you do this? Well, you have to copy and paste your figure from this file here, right? Copy and paste it, fix it up, you know, trim it, you know, playing with, sorry, I didn't have that on the screen. So playing with the some of the image corrections can be helpful because, you know, in the end, what you really want are the darkest bands, right? Like, you know, these bands here, which are barely bands, we don't really care about. That, you know, you could barely call that a band 2. I really want just the darkest bands, that band, that band, that band, you know, maybe that band. This is going to be subjective, okay? And you want to be consistent from sample to sample. Like, I wouldn't want to say, you know, this is a band in this sample, but not in this sample, right? So, you know, I just want the darkest bands for you to include in your analysis. But you have to print it, and you have to decide your bands and do your measurements on a printed page. You cannot do this on the computer screen with a ruler. You know, just don't do that. You have to print the thing and then whip out a ruler and start taking measurements. Okay. All right. With that being said, I'm going to delete this. And I know some groups are saying, no, because I was going to use that one. Oh, you're going to have to do it yourself. And I'm going to use this as an example moving forward. All right, so I've printed this and I've labeled some things here. First, I went in, well, first thing, so when you have like a fat band like that, generally what people do is to actually, like, where they measure the band is at the bottom of the fat band. So if you can see, I'm just trying to look at a good example here. Let's blow that up. You can see, like, okay, so this band here, it takes up all of that area there, but you just draw a line at the bottom of the bands that you're measuring so that you know at what point to read on the ruler. That's how you decide where, because the bands are, some of them are pretty thick. All right, so in an area above all of the bands, you're going to draw a line. This is your beginning point. And in an area below all of your bands, you're going to draw a line. This is your end point. And you're going to measure the distance on paper between the beginning and end point. And I've measured here 91 millimeters. You're going to do this all in millimeters so you can get some precision here between different bands. Okay. Now, we're only analyzing bands of the lower molecular weight. So the molecular weights that you're going to be analyzing are between 10 and 30 kilodaltons. So what I've done is I've drawn a line here between 30 and 10 kilodaltons so that we're just going to focus on the bands that are in this range. Okay. Now, the standard is what we're going to use to generate our standard curve. And we're going to use the standard curve to find the molecular weights of all of these other bands. I have only done the first two lanes here as an example. So you have more bands here that you need to calculate for, right? There are bands, I see 3 bands there, another band here, 2 there, 1 there, 1 there, 1 there, 1 there, and so on, okay? For the first part, the SDS page, you can ignore your actin myosin standard. You don't need that. But you see, I've got this crossed out up here because we don't care about any of these larger molecular weight bands. Right? You really can only use the lower molecular weights to kind of generate proteomic profile because if you see, you notice these are they all look the same, right? All right, so now I did include with the standard, I went ahead and include the 50 and the 37. What did I write? Oh, yeah. The 50 and the 37 kilodalton marker here, because there are some bands up here in shark and salmon and also in trout and sturgeon that are between 25 and 37. So in order to be able to, that those can fit in the linear range of the curve, we need to include that. All right, I'm including this 50 kilodalton one. You don't have to, it's optional, but I'm going to go ahead and include it. Okay, so now I have measured from the origin to each of these bands. I've gone from here to here, that's 17 millimeters, and here to here, that's 27 and 39, 48. Down to this one, there's a band there, it's 81. And I've done the same thing for all these bands here. Okay, that's why you have to do this on paper, and you draw where you're going to measure to, and you make your measurements. All right? Again, it's from the origin to where the band is. Okay, so now we're going to generate our standard curve. So the standard curve is going to plot molecular weight, which is going to go 50, 37, 25, 20, 15, and 10 versus RF. Okay, RF is relative mobility. And to generate that, we have to take the distance migrated by the band, which is here, 17, 27, 39, 48, 62, 81, and divide it by the distance migrated by the die front, which is 90, no, it's 91 for each sample. Okay. So this isn't confusing here. So this is going to be this divided by this. And your value should always be less than 0.1. Okay. Now we're going to plot this molecular weight versus our RF. Oops. Paste the value, not the formula. And I'm going to select those, and we're going to go up to Insert, and we're going to do a chart. It's the dot plot, so this one. And you want to make sure that molecular weight is on the, molecular weight is on the y-axis, which you can see here, it's not. So I'm going to just delete this. I'm going to switch these columns here. Cut, paste, and now it should do it correctly. I always forget to do that right. So insert, plot, dot plot, this guy, and that looks correctly. So here we have our molecular weight, here we have our RF. Okay. Now we need to generate an equation for this. All right, so we're going to click on any point. We're going to right click. We're going to go to add trend line. We need to do exponential, and we need to do display equation on chart and display R squared value on chart. Okay. Now, this is not correct as is, because in order to do exponential regression analysis correctly, what you're regressing here has to be in logarithmic format. So I'm going to make sure sometimes I get this wrong here. Format axis and put it to logarithmic scale. Voila, it did it. That worked. Okay, so your y-axis, which is your molecular weights, not RF, needs to be logarithmic, and that's how you do it. Right click, you go format axis, and you do that. Okay. All right, so there is our equation. Now, we need to use this as a tool to find the molecular weights of all of our bands. Okay. And in order to do that, you have to now find the RF value for all of those bands and then plug it into the equation here and solve for Y, right? So you solve for Y after you plug those RF values. So, right, it's just like we did before, right? You're taking this value that you measured, divided by the die front, 91. So let's do shark here. So we've got, I think I can read my writing there, 33, 40, 45, 56, 64, 67. And these are all going to get divided by 91. And that is going to equal this divided by this. Okay. And you can use Excel to work with that formula as well. I'm not going to tell you guys how to do that. I want you guys to figure that one out. All right. You don't have to use Excel to do that. You can use your calculator. But I will tell you that since you're doing, remember this was exponential. Okay. You have to use the natural log function to calculate it. Right? So you're just taking this value here, plugging it into this formula for X, and solving for Y. But in order to do that E to that on your calculator, use the natural log, which is the LN button on your calculator. Okay. You can also, use Excel to do that. I'm not going to show you how to do that. You can look that up. Okay. All right. So, what do you get from all this? Well, now you have the molecular weights for all of these bands here. Okay. So you're going to have, 1, 2, 3, 4, 5, 6, about 30, 2, 3, 4, 5. Yeah, I'd say about 30, 35 values. All right. And at this point, we're done with our standard curve. We're just using the standard curve as a tool to find these molecular weights. I'm going to go ahead and save this file and upload it to Canvas so that you guys can reference it. If you need to look up the exponential or anything like that. Proteomics lab. This is standard curve calculation. Okay. All right. Okay. So what do you do after you get all those values? Well, you're going to basically put all those values. Oh, also that standard curve is going to be one of your figures. I want you to copy and put that figure in. It needs a legend. It needs a title. Let's see, where are we here? So standard curve. Yeah, include this curve as your next figure. This is figure 3. Okay. But we use that curve to generate all of those values of all the fish species, lower molecular weight between 10 and 30 kilodaltons. So that needs to find its way into a table. And I'm going to show you how you're going to generate that table. If you look on page 78 of Student Manual 1, I have the thing open in, there's my Adobe. There we go. 78. There we go. So you're going to have this big table where you've listed out all of your species, right? And then, so let's say the shark or whatever it was. Where's my file here? Oh, open that back up. Oops, sorry. All right, so if you see for shark, we calculated 1, 2, 3, 4, 5, 6. 6 bands. That means that you're going to have like 6 X's here for that species. All right. Now this one did 8. Okay, might have done a little differently. It's fine. All right. But so you can see the way this table works is you've got all of the molecular weights of all of the bands, right? And it's including the distance migrated there, right? But then it shows each band, for each species. So in other words, in species A, it had a band that's 21.7 kilodaltons. And look, it's the only species that had that band. But, the 28.6 kilodalton band, 4 species had it, right? So this kind of table can start to make comparisons, right? And then on the table, you count up the number of bands that each species had, and you put it down here. Okay, that's the total number of bands that each species had. This is going to be the next table in your lab report. All right. Then you're going to take that data and you're going to use it to generate a new table where you list the bands that each species, you list the bands that each species has in common, right? And so here you've got, so species A had 8 bands, right? With species A. So species E had 12 bands. You can see like the numbers, how they're going here. 8, 10, 13, 10, 12 is the same here. 8, 10, 13, 10, 12. All right, but the number of bands that 2 species have in common is listed here, right? So, and you need to reference this table that you generated and use it to compile a table here where you could say species C and A have 2 bands in common. Okay, so now you can see you're starting to get data that shows which species have more in common than others, right? And the instructions here, so this is going to be another table in your lab report. Okay, it's going to be table 2, 3. Okay. Both these tables, put them in your results. Remember, tables get a descriptive title and appropriate column and row headings. All right, and then the instructions in here are going to walk you through how to generate a cladogram. Okay, now these are not complex drawings. Okay, it's just lines, and you should be able to generate this like in PowerPoint or Word. I recommend PowerPoint; it's a little easier, to do that. Okay, but make a digital copy. I really don't want you guys drawing this out on paper. I want you to take the time to figure out how to do it in digitally. All right. So, but these instructions will tell you how to do this, and basically, where there's divergences between different species, it will show you how to do that. So in the end, you will have a cladogram. Right. So here, species C and B are closer together on the branching, right? So they're closer together evolutionary. Okay. That cladogram is going to be a figure in your lab report. That is going to be figure 4. Okay. All right. That pretty much concludes the first component, just the SDS page, Kamasi component where you're generating the cladograms. Okay. All right. The next part is going to focus on the Western blotting stuff. Okay. So with Western blotting, what we're doing is just looking at 2 proteins, which are myosin light chain 1 and myosin light chain 2. Those were the proteins that we probed for on our Western blot. So here you can see, I think light chain 1 is the smaller one. The bottom, light chain 2 is the bigger one, I believe. I might be reversing those. But so you can see this one here just has, I don't know if that's 1 band. It's really hard to see. I would need to zoom that up. Zoom that in. It could be 2. But it looks like it just has 1 there, myosin light chain 1. Okay, so what you're going to do is you're going to end up doing a very similar thing. You're going to generate another standard curve for this. Okay. And you're going to find the molecular weights of these different bands here. And then you're going to compare them to the actual molecular weights, right? So these would be your experimental results. And then now we're going to also generate the empirical results. Okay. So if you look at pages 90 to 92 in Student Manual 2, we're going to use that 90 to 92. Uh-oh. Something like this. Yeah, so 90 to 92 is going to explain to you like the about how to get these sequences. So I'm going to walk you through that a little bit here. Okay, so to get the sequences, we need to go to NCBI protein. All right. And when you go to NCBI protein, you're going to drop down, you're going to pick protein, and you're going to search for myosin light chain 1, Salmo salar. Now, where are you getting these species names from? Your fish data sheets, right? Okay, and this pulled up nice because I walked through this before just to check it. So now sometimes a screen will pop up that like has all your options again, but it shouldn't as long as you select a protein here. All right, and I asked you to look at this session number explicitly. So 7085, which is the first one. So generally it's going to be the first one. So And you will be able to find myosin light chain for most of these fish species, but I can't guarantee it. Some of them you may be missing some of the results. Okay? Um, and typically it's the first one that comes up is the one you want. But for each of your fish species, you're going to search for myosin light chain 1 and the fish species name, and then you're going to go in there and get myosin light chain 2 and the fish species name. Okay, so 5 fish species, 2 light chains. You've got 10 sequences that you should be able to retrieve, but 1 or 2 may be missing. We're at the limitation of if some of the species are more studied than others. Okay. Anyways, you pull it up, and we're going to be able to get the amino acid sequence, which is right down here. All right. I recommend copying that, pasting it to a Word document so you can start organizing them. You do not need to present these sequences in your report. Okay. We're going to copy this guy, and we're going to go to another website called Xpassi. And Xpassi has all these online computational tools. We're going to paste the sequence in there, and we're going to use that to compute the theoretical molecular weight, which is right here. And this is PI. PI is your isoelectric point. We don't need to worry about isoelectric point because we didn't do any 2D gels. But so this is the molecular weight in Daltons, right? So everything so far has been in kilodaltons. So this would correspond to about 21.01 or 02 kilodaltons. Okay. So for both light chains 1 and 2 and all 5 fish species, you need to do this. And you need to get this value, the molecular weight, and you need to construct a table, okay, that is going to compare those theoretical weights versus the empirical molecular weights that you got from generating another standard curve of this image, right? So, just like we did before, but you're going to need to print this off and do it as a separate image because this was a separate gel. Your numbers are going to be slightly different. So print this off separately, get a origin and a die front distance, calculate RF, all that stuff. Okay, just like I taught you before, and then get a empirical molecular weight for each of those bands. And then you're going to construct a table where you compare the empirical versus theoretical data. Okay, that's going to make up figures. Sorry, I'm down here. Figures 5 and table 4. Is that right? Too? So that's where my error is. I got to fix that. It's actually table 5. I was missing something. And I just want to double check this down here. 5 tables, fish predicted. So what's one I'm missing here? Fish summarize data table, band comparison, band similarity, predicted versus empirical weights. Sorry guys, just give me a sec here. I want to figure this out. Table 5. Figure 4. Oh, no. I keep screwing this up. This is table 3. This is table 4. It's not 5 tables. That was right. Bam. That was there right there. Okay. All right. You can ignore all that. So it's just saying. All right. So now all this work has been done by everyone in the group, working together to generate these 4 tables and 5 figures. Right. 5 figures and 4 tables. And everyone's going to be the same. The legends can be the same. I don't care about that. Titles can be the same. And now you've got those 5 figures and 4 tables, and you're off on your own. And now you're going to write up your lab report by yourself. Okay. All right. Everyone's probably familiar with what scientific manuscripts look like. I put one of my most recent. Which one is this? 223. This is not my most recent. I had one come out earlier this year. But this is one of my papers from previous work. So you can use it as an example if you're able to get this printed off. And all I want you to use this to do is to kind of see what is included in each section, right? So generally what you include, you have a descriptive title, right? It's almost a full sentence, but not. It's not just like, lab report 1, right? Or fish analysis, right? It's a descriptive title, summarizes the entire paper. Generally, my recommendation is you're going to generate this last. This is the last thing you're going to do before you print it off is put your title in, because I think after writing everything up, you can safely develop a title. The second to last thing you're going to do is generate your abstract, because after writing up all the sections, then you can now generate a summary of all of those sections together. Okay. And that's why, importantly, I've recommended the, I don't have title in there. I should probably add it. The order, when you're writing your paper, this is the order you should write it in. Start with your results. You've already got the figures. Now you just need to write the text of your results, because the results is written in prose. If we look at this paper here, sometimes it's easier to look at a PDF. I don't know if I can download that. It's going to make me log in. Nope. Computers. Where did that go? Disappeared. There it is. Results. See, the results has prose writing in it, right? And it references the figures. Okay, and it's chronological. Typically, it's chronological because it's telling the story, right? So you can see so far. You can see how we've developed all the figures and the order that they should be in. All right, and there are some details that I've kind of mentioned that maybe 100% don't fit this paper. Like most results sections, sometimes results are written up with subsections, okay? And you could do that. You can have subsections. But you can see here this figure has a nice figure legend. And if there's any data tables in this paper, I don't remember offhand. There aren't actually. So, but tables will not have legends. It'll just have the title and thing. Okay. Also, it's useful in the materials and methods to kind of see, what sort of details are included and excluded, right? But I'm not requiring that you read this entire paper, understand this entire paper. It's just as a guide, so you kind of get an idea of what sort of stuff you're going to include in your report. Okay, so when you're writing a report, write it in this order. I'm going to include title down there. And it will make it so much easier to do. Okay, but right, this is I just don't know why this is an area of so much confusion for students, but it is. It is not the order that you're going to present the report in. Okay, so after you write it up, you're then going to rearrange the sections into the correctly presented order, which is how it's given in this paper, which is title, abstract, intro, materials and methods, results, discussion, literature cited. Okay, but when generated in this order will make it a lot easier. Okay, so I kind of walked through here in more details. I'm going to start wrapping this up here, guys, because I got to go. But so, the details to include the figures, the tables, and then the writing. So results is going to have writing. And this part needs to be different for everybody, right? This is your lab partners are not doing this as a collaborative effort. Okay. This is written in prose. It cites the tables and explains all the data. All right. Introduction, approximately 1 page, and that's not double spaced, right? So I've given you some suggested topics here. Hopefully, also, as you've answered all those focus questions, you can kind of get an idea of all the stuff that we're talking about here, right? Muscle proteins, myosin, Western blotting, SDS page. The purpose of the introduction is to provide background information for the reader so that he can understand the rest of the paper. But you're not writing this to a third grader, okay? This is another college student. So, we're not going to explain what a cell is and give background information on a cell, right? We're going to assume they know what a cell is. We assume they know what a protein is, things like that. Your introduction is going to include your hypothesis there, so usually generally towards the end, but it doesn't explicitly say, I hypothesize that, right? It just kind of, it mentions it in there in, we predict, something like that, right? No papers actually write that, I hypothesize. Okay, but your hypothesis is, which fish species were in common and which were not. Okay, materials and methods. This should be close to a page. It is divided into subsections. All right, for example, we extracted the protein, then we ran the gel, then we did the Western blotting. But there's other stuff here too, because you need to provide, how the standard curve was generated. And you need to be sure the websites are cited, that you went to Expasi to do calculate the molecular weight. So, this is not everything. It's written in third person. It needs to include the details that are necessary for someone to recapitulate the experiment. So, the antibody concentration and the incubation times, those are very important because they will affect results. Those need to be the same. But, things like I turned on the machine. Okay, come on. We don't need that. That's not unnecessary information. All right. The discussion, right, analyzes each experiment. Okay. So, and this is the main thing. Do your results support your predictions? If they don't, why? If they do, great. Why? Don't go crazy on this because students love to do this, and I don't really like reading about this stuff. I'm gonna be honest with you. We could have ran it at a different voltage or something like that. And I don't, get into it a little bit, address it, but don't go too crazy with it. Improvements, right? The things to discuss here, besides your hypothesis, are things that were addressed in these questions should be a good guide of things that should be included. Okay. The abstract is basically a one concise paragraph that brings together all of the sections, right? That's why you want to write it last. The discussion is bringing together, your results and your introduction, right? So that's why you want to write this section after writing these sections. Okay. I think you guys get it. All right. So informative title, concise title, and then your references are in numerical formats. You're going to reference things like this. So how many references? I don't know. And you're going to use the Biorad manual, obviously. I would like you guys to get at least, use at least two references excluding your textbook and these manuals. That could be another scientific report, probably something on, you want to pull up something on fish muscle or on fish species, and pull up something maybe on Western blotting. Okay, so just two outside sources. When you cite them in the introduction and the discussion, you're going to do like in parenthetical citations, right? Where do I go over that? Intro. Oh, that's right. Yeah. So after each sentence, you write the last name of the first author, which was not me, 2010. All right, that's just an example. So, so whatever, like if you're citing this, it's going to be Lou, and it's going to be the year that it was published, and that's it. You don't need to put the page number or anything like that. Just the article that it was pulled from. And your introduction, for example, every line in that should pretty much be cited, right? I mean, I doubt you're using your own information in anything in the introduction. You're getting it all from the manual or from outside sources, except your hypothesis. That should be the only couple sentences that aren't cited in your intro. The discussion will have some citations, but less because it's going to be more your own, your own thoughts and whatnot. But all this stuff is just background information. And I don't say that here, but I should. Pretty much every line should be cited. All right. So that 6-page total, that's actual writing to generate all of these sections, including in your results, the prose that kind of brings together all the figures. It doesn't include the figures. Your whole report's probably going to be 12 pages, including all the figures. All right. But I want these figures nice. The tables should be tight and neat. They should fit on the page. If they're running off on pages and it's sloppy and it's difficult to grade, that's gonna be reflected in the grade that you receive. Okay. All right. So we're gonna meet next week. You've had time here. I've gone over what's required and expected for this, and you'll have questions, and that's fine. Please bring me any questions. And good luck in generating your lab report. All right. I will see you guys soon. And that is it for this.